Bacteria are microscopic, single-celled organisms that lack a nucleus and other membrane-bound organelles, placing them among the simplest known forms of life. They are classified as prokaryotes, meaning their genetic material is not enclosed within a nuclear membrane, but exists as a single, usually circular DNA molecule in the cytoplasm. Despite this apparent simplicity, bacteria are structurally and biochemically sophisticated and are capable of carrying out all essential life processes independently. They inhabit virtually every environment on Earth, including soil, water, air, the deep subsurface, polar ice, hydrothermal vents, and the bodies of plants and animals. Their ability to survive and thrive across such diverse conditions makes them one of the most successful life forms in Earth's history. The importance of bacteria lies primarily in their fundamental role in sustaining life on the planet. They are indispensable participants in global biogeochemical cycles, including the carbon, nitrogen, sulfur, and phosphorus cycles. Through processes such as decomposition, nitrogen fixation, nitrification, and denitrification, bacteria regulate the availability of essential nutrients, enabling ecosystems to function. Without bacterial activity, organic matter would accumulate without being recycled, and many elements necessary for life would become biologically inaccessible. These processes operate continuously and largely invisibly, forming the biochemical foundation upon which plants, animals, and entire ecosystems depend. Bacteria also play a critical role in the origin and evolution of life. Fossil and molecular evidence indicates that bacteria emerged more than 3.5 billion years ago, making them among the earliest life forms on Earth. For most of Earth's history, bacteria were the dominant and often the only organisms present. Some bacterial lineages developed oxygen-producing photosynthesis, leading to the accumulation of oxygen in the atmosphere during the Great Oxidation Event. This transformation fundamentally altered Earth's environment and made the evolution of complex, oxygen-dependent life possible. In this sense, modern ecosystems and multicellular organisms exist largely as a consequence of ancient bacterial activity. In modern biology and medicine, bacteria are central to both health and disease. Many bacterial species live in close association with humans and other organisms, forming complex microbial communities that influence digestion, immune development, metabolism, and protection against pathogens. These beneficial bacteria are essential for normal physiological function. At the same time, a relatively small fraction of bacterial species can cause infectious diseases by invading tissues, producing toxins, or triggering harmful immune responses. Understanding bacteria is therefore essential for diagnosing, preventing, and treating disease, as well as for managing antibiotic use and combating antibiotic resistance. Beyond natural ecosystems and health, bacteria are vital to human technology and industry. They are widely used in food production, biotechnology, agriculture, environmental management, and scientific research. Bacteria synthesize enzymes, vitamins, antibiotics, and bioactive compounds, and they serve as model organisms for studying fundamental biological processes such as gene regulation, metabolism, and evolution. Their rapid growth, genetic adaptability, and biochemical diversity make them powerful tools for innovation. Overall, bacteria are not merely microscopic organisms of academic interest. They are foundational to life, deeply integrated into Earth systems, and central to humanity's past, present, and future. The discovery of bacteria is closely tied to the development of microscopy in the 17th century. Before this period, bacteria were entirely unknown because they are far below the resolution of the unaided human eye. The first recorded observations of bacteria were made in the 1670s by Antony van Leeuwenhoek, a Dutch tradesman who constructed powerful single-lens microscopes. Using these instruments, he observed microscopic organisms in water microscopic work by Robert Hooke laid important groundwork, but did not include true bacterial observations. Hooke's 1665 book Micrographia documented microscopic structures such as plant cells and molds, demonstrating that living organisms could exist beyond the limits of human vision. However, Hooke's microscopes lacked the resolving power necessary to clearly visualize bacteria. Hooke's technical improvements, particularly his ability to grind lenses with high magnification and clarity, marked a decisive step forward in revealing the microbial world. For more than a century after Hooke's discoveries, bacteria remained largely a scientific curiosity. Their biological significance was not fully understood, and debates persisted about their origin and role in nature. One major controversy concerned spontaneous generation, the idea that living organisms could arise from non-living matter. This view was challenged and ultimately refuted in the 19th century through a series of controlled experiments, most notably by Louis Pasteur. Pasteur demonstrated that microorganisms, including bacteria, originated from pre-existing life rather than appearing spontaneously, establishing a core principle of microbiology. The 19th century marked the formal birth of microbiology as a scientific discipline. Advances in microscopy, staining techniques, and experimental methods allowed scientists to study bacteria systematically. Pasteur also showed that bacteria were responsible for fermentation and spoilage, linking them to specific biochemical processes. These findings overturned earlier chemical explanations and established bacteria as living agents with distinct metabolic activities. Pasteur's work further demonstrated that certain bacteria could cause disease, providing early evidence for what would later become the germ theory of disease. The germ theory was solidified by the work of Robert Koch, who developed rigorous methods to link specific bacteria to specific diseases. Koch introduced techniques for culturing bacteria on solid media, allowing individual species to be isolated and studied in pure form. He also formulated criteria, later known as Koch's postulates, to establish a causal relationship between a microorganism and a disease. Through his work on diseases such as anthrax, tuberculosis, and cholera, Koch demonstrated conclusively that bacteria could be primary agents of infection. By the late 19th and early 20th centuries, the study of bacteria had expanded rapidly. Classification systems were developed based on morphology, staining behavior, and metabolic traits. The invention of gram staining provided a powerful method for distinguishing major bacterial groups based on cell wall structure. As microbiology matured, bacteria came to be recognized not only as causes of disease, but also as essential components of natural processes. The discovery of bacteria thus transformed biology, medicine, and chemistry, revealing an unseen majority of life that profoundly influences the living world. Bacteria occupy one of the three primary domains of life, a classification that reflects deep evolutionary relationships rather than superficial similarity. Modern biological taxonomy divides all cellular life into bacteria, archaea, and eukarya. This domain-based system emerged from molecular comparisons of ribosomal RNA sequences, which revealed that bacteria form a fundamentally distinct lineage. Unlike eukaryotes, bacterial cells lack a nucleus and membrane-bound organelles, and unlike archaea, their cellular membranes, gene transcription machinery, and many metabolic pathways follow different biochemical patterns. Placement within the domain bacteria reflects shared ancestry and conserved molecular traits rather than ecological role or morphology. The recognition of bacteria as a separate domain is largely attributed to the work of Carl Woese in the late 20th century. By comparing 16S ribosomal RNA sequences, Woese demonstrated that prokaryotic life was not a single unified group, but instead consisted of two deeply divergent lineages, bacteria and archaea. This discovery overturned the long-standing two-kingdom and later five-kingdom classification systems, which grouped all prokaryotes together. Molecular phylogeny showed that bacteria share a common ancestor distinct from that of archaea and eukaryotes, reshaping understanding of early evolution. Within the bacterial domain, diversity is immense. Bacteria are divided into numerous phyla based on genetic, biochemical, and structural characteristics. These phyla include lineages that differ widely in cell wall composition, metabolism, ecological function, and evolutionary history. Some bacterial groups are closely related, while others diverged billions of years ago. This genetic diversity exceeds that found within plants or animals, reflecting the long evolutionary history and rapid adaptability of bacteria. Many bacterial lineages are known only from genetic sequencing and have never been cultured in the laboratory. From an evolutionary perspective, bacteria represent some of the earliest branching forms of life. Evidence from isotopic signatures and fossilized microbial structures indicates that bacterial ancestors existed more than 3.5 billion years ago. Early bacteria evolved in environments lacking oxygen and relied on anaerobic metabolic pathways. Over time, bacterial evolution produced extraordinary metabolic innovation, including photosynthesis, sulfur metabolism, iron oxidation, and methanogenesis-related processes in associated lineages. These innovations allowed bacteria to colonize nearly every ecological niche on Earth. The relationship between bacteria and other domains of life is central to understanding cellular evolution. Eukaryotic cells are thought to have arisen through endosymbiosis, a process in which ancestral bacteria entered into long-term associations with larger host cells. Mitochondria and chloroplasts retain bacterial features such as circular DNA, bacterial ribosomes, and division by binary fission, indicating their origin from bacterial ancestors. This evolutionary event means that modern eukaryotic life is fundamentally dependent on bacterial lineages that became integrated into complex cells. In the tree of life, bacteria are not a peripheral or primitive group, but a foundational domain that shaped the planet's biology. Their early diversification established metabolic pathways that continue to sustain ecosystems, and their evolutionary flexibility allows them to respond rapidly to environmental change. Understanding the place of bacteria in the tree of life provides insight into how life began, how it diversified, and why bacteria remain the most abundant and genetically diverse organisms on Earth. Bacterial cells exhibit remarkable diversity in size, shape, and structural organization. Despite sharing a basic prokaryotic plan, most bacteria are extremely small, typically ranging from about 0.5 to 5 micrometers in length. Although some species fall outside this range, their small size results in a high surface area to volume ratio, which enhances nutrient uptake and waste removal, and contributes to rapid growth under favorable conditions. This microscopic scale also allows bacteria to inhabit environments inaccessible to larger organisms, including thin water films, soil pores, and internal host tissues. The most commonly observed bacterial shapes are cocci, bacilli, and spirilla. Cocci are spherical or nearly spherical cells that may occur singly, in pairs, chains, clusters, or more complex arrangements, depending on how cell division occurs and whether daughter cells remain attached. Bacilli are rod-shaped bacteria, which can vary greatly in length and width and may appear singly or in chains. Spirilla and spirochetes exhibit helical or corkscrew-like forms, which can aid motility in viscous environments. Additional shapes include curved rods, filamentous forms, and pleomorphic bacteria that lack a consistent shape. Cell shape is not arbitrary, but is determined by the structure of the bacterial cell wall and cytoskeletal elements. The rigid cell wall, primarily composed of peptidoglycan in most bacteria, maintains structural integrity and in cell wall thickness and composition, influence cell shape and mechanical strength. Internally, bacterial cytoskeletal proteins help coordinate cell division, maintain polarity, and guide cell wall synthesis. These systems allow bacteria to maintain defined shapes while still being capable of growth and division. Beyond basic shape, bacterial cells display structural diversity through specialized surface features. Many bacteria possess flagella, which are long, whip-like appendages that enable movement. Flagella can be arranged in different patterns, such as a single flagellum at one pole, multiple flagella at one or both poles, or numerous flagella distributed over the cell surface. Other bacteria have pili or fimbriae, which are shorter, hair-like structures involved in attachment to surfaces, host cells, or other bacteria. These structures play important roles in colonization, biofilm formation, and genetic exchange. Some bacteria exhibit extreme structural adaptations that reflect their ecological niches. Filamentous bacteria form long chains of cells that resemble fungal hyphae and are well suited for growth in soil environments. Certain aquatic bacteria are unusually large, enabling them to store nutrients or avoid predation. A few bacterial species possess internal membrane systems that compartmentalize metabolic processes, challenging the traditional view that all bacteria lack internal organization. These variations demonstrate that bacterial structure is far more complex than once assumed. Structural diversity among bacteria is closely linked to function and survival. Shape influences how bacteria move, attach to surfaces, resist physical stress, and interact with hosts or other microorganisms. Differences in size and morphology affect susceptibility to predation, efficiency of nutrient uptake, and ability to form multicellular communities. Collectively, the wide range of bacterial sizes, shapes, and structures reflects billions of years of evolution and adaptation, allowing bacteria to occupy nearly every environment on Earth and perform an extraordinary variety of biological roles. Bacterial cell walls and membranes are essential structural components that define cell shape, protect against environmental stress, and regulate interactions with the surroundings. The cell wall provides rigidity and prevents osmotic lysis in hypotonic environments, while the cell membrane functions as a selectively permeable barrier that controls the movement of ions, nutrients, and waste products. Although all bacteria possess a cytoplasmic membrane composed of a phospholipid bilayer with embedded proteins, the structure and composition of the cell wall vary significantly among different bacterial groups. One of the most important distinctions in bacterial cell wall structure is between gram-positive and gram-negative bacteria, a classification based on their response to the Gram staining procedure. Gram-positive bacteria have a thick, multi-layered cell wall composed primarily of peptidoglycan, a polymer of sugars and amino acids that forms a rigid mesh surrounding the cell membrane. This thick peptidoglycan layer retains the crystal violet stain during Gram staining, causing these bacteria to appear purple under a microscope. Embedded within the peptidoglycan are teichoic acids and lipoicoic acids, which contribute to cell wall stability, ion regulation, and interactions with host tissues. Gram-negative bacteria possess a more complex cell envelope, consisting of a thin peptidoglycan layer located between two membranes. Outside the peptidoglycan lies an outer membrane that is unique to gram-negative bacteria. This outer membrane contains lipopolysaccharides, phospholipids, and proteins, forming an additional protective barrier. During Gram staining, the thin peptidoglycan layer does not retain the crystal violet stain, and the cells take up the counterstain instead, appearing pink or red. The presence of the outer membrane significantly influences how these bacteria interact with their environment and respond to antimicrobial agents. The lipopolysaccharide component of the gram-negative outer membrane plays a critical biological role. It consists of lipid A, a core polysaccharide, and an O-antigen side chain. Lipid A anchors the molecule in the membrane and can trigger strong immune responses in animals, while the O-antigen varies among species and strains, contributing to antigenic diversity. The outer membrane also contains porin proteins that form channels allowing small molecules to pass into the periplasmic space. This selective permeability affects nutrient uptake and limits the entry of toxic substances, including certain antibiotics. Between the inner membrane and the outer membrane in gram-negative bacteria lies the periplasmic space, which contains the peptidoglycan layer and a variety of enzymes and binding proteins. This compartment is involved in nutrient processing, detoxification, and assembly of cell wall components. Gram-positive bacteria lack a defined periplasmic space, but may contain functionally similar enzymes associated with the cell membrane or embedded within the thick peptidoglycan matrix. These structural differences have major implications for physiology, pathogenicity, and antibiotic susceptibility. The cytoplasmic membrane in all bacteria is a dynamic structure composed mainly of phospholipids and proteins. It serves as the site of energy generation, including electron transport and proton gradient formation. Because bacteria lack mitochondria, transport proteins embedded in the membrane actively import nutrients and export waste, while sensory proteins detect environmental signals. Together, the cell wall and membranes form an integrated system that maintains cellular integrity, enables metabolic activity, and underpins the survival and adaptability of bacteria across diverse environments. The bacterial cytoplasm is the aqueous interior of the cell enclosed by the cytoplasmic membrane and contains the components necessary for metabolism, growth, and reproduction. It is a highly crowded and organized environment rather than a simple fluid. The cytoplasm consists primarily of water, dissolved ions, metabolites, enzymes, nucleic acids, and structural proteins. Its composition is tightly regulated to maintain appropriate pH, ionic strength, and redox conditions, all of which are essential for enzymatic reactions and cellular stability. Bacterial ribosomes are among the most abundant structures in the cytoplasm and are responsible for protein synthesis. They are smaller than eukaryotic ribosomes and are classified as 70S ribosomes, composed of a 50S large subunit and a 30S small subunit. These ribosomes translate messenger RNA into polypeptide chains by sequentially adding amino acids. Because bacteria often grow rapidly, ribosomes can make up a large fraction of the cell's mass. The structure and function of bacterial ribosomes differ sufficiently from eukaryotic ribosomes that many antibiotics selectively target bacterial protein synthesis without affecting host cells. In addition to ribosomes, the cytoplasm contains the bacterial chromosome, typically a single, circular DNA molecule localized in a region called the nucleoid. Unlike a nucleus, the nucleoid is not surrounded by a membrane, but the DNA is highly compacted and organized through supercoiling and association with DNA-binding proteins. This organization allows efficient replication, transcription, and segregation of genetic material during cell division. The close physical proximity of DNA and ribosomes enables transcription and translation to occur simultaneously, a feature unique to prokaryotes. Bacterial cytoplasm often includes plasmids, which are small, circular DNA molecules independent of the main chromosome. Plasmids carry genes that can confer advantageous traits such as antibiotic resistance, metabolic capabilities, or virulence factors. They are replicated separately from the chromosome and can be present in multiple copies. The presence and maintenance of plasmids contribute significantly to bacterial adaptability and genetic diversity, especially in changing environments. Many bacteria contain cytoplasmic inclusions, which are discrete structures used for storage or specialized functions. These inclusions may store carbon, nitrogen, sulfur, or phosphorus in polymerized forms that can be mobilized when external nutrients are scarce. Examples include glycogen granules, polyphosphate bodies, and lipid inclusions. Some bacteria possess gas vesicles, protein-bound structures that provide buoyancy and allow cells to position themselves optimally in aquatic environments. Although bacteria lack membrane-bound organelles, their cytoplasm is functionally organized through protein complexes and localized biochemical pathways. Enzymes involved in related metabolic processes are often spatially associated, increasing reaction efficiency. Certain bacteria also contain protein-based microcompartments that enclose specific enzymatic reactions, reducing interference with other cellular processes. These structures demonstrate that bacterial cells achieve compartmentalization through mechanisms distinct from those of eukaryotic cells. The bacterial cytoplasm is therefore a dynamic and highly regulated environment that supports all essential cellular activities. Its organization allows bacteria to grow rapidly, respond quickly to environmental changes, and survive under diverse conditions. Far from being structurally simple, the cytoplasm reflects an efficient evolutionary design that maximizes functionality within minimal cellular space. The genetic material of bacteria is organized in a fundamentally different way from that of eukaryotic cells. Most bacteria possess a single chromosome composed of double-stranded DNA that is typically circular. This chromosome contains the majority of genes required for cellular structure, metabolism, replication, and regulation. The DNA is densely packed into the nucleoid region of the cytoplasm through supercoiling and association with DNA-binding proteins, allowing large genomes to fit within very small cellular volumes while remaining accessible for transcription and replication. Bacterial genomes vary widely in size and gene content. Some bacteria have compact genomes with fewer than 1,000 genes, while others possess several thousand genes encoding diverse metabolic and regulatory functions. Genome size is closely linked to lifestyle. Obligate parasites and symbionts often have reduced genomes due to reliance on host resources, whereas free-living bacteria typically maintain larger genomes that support metabolic flexibility. Gene density in bacterial chromosomes is high, with relatively little non-coding DNA compared to eukaryotic genomes. In addition to the main chromosome, many bacteria carry plasmids, which are smaller, extrachromosomal DNA molecules capable of independent replication. Plasmids are usually circular and may exist in single or multiple copies within a cell. They are not essential for basic survival under all conditions, but they often encode traits that provide selective advantages, such as antibiotic resistance, toxin production, or the ability to metabolize unusual compounds. Plasmids can be gained or lost without killing the cell, making them powerful agents of rapid adaptation. Gene regulation in bacteria is highly efficient and tightly controlled. Genes that participate in the same metabolic pathway or functional process are often organized into operons, which are clusters of genes transcribed together as a single messenger RNA. This arrangement allows coordinated regulation of related genes in response to environmental signals. Regulatory proteins can activate or repress transcription by binding to specific DNA sequences near promoters, enabling bacteria to conserve energy and resources by producing proteins only when they are needed. Bacterial gene expression is closely linked to environmental conditions. Changes in nutrient availability, temperature, pH, oxygen concentration, or stress factors can rapidly alter transcriptional and translational activity. Because transcription and translation occur simultaneously in bacteria, regulatory responses can be extremely fast. This coupling allows bacteria to respond almost immediately to external changes, a critical advantage in fluctuating environments. Genetic variation in bacteria arises through mutation horizontal gene transfer. Mutations occur due to errors in DNA replication or damage, caused by environmental factors such as radiation or chemicals. Although most mutations are neutral or harmful, some provide beneficial traits that can be selected for under specific conditions. Horizontal gene transfer allows bacteria to acquire genes from other organisms rather than relying solely on inheritance from parent cells, greatly accelerating evolutionary change. The organization and regulation of bacterial genetic material support remarkable adaptability. Compact genomes, operon-based regulation, plasmid-mediated traits, and rapid gene expression enable bacteria to survive diverse challenges and exploit new ecological niches. These genetic features explain how bacteria can evolve resistance to antibiotics, colonize extreme environments, and maintain dominance as one of the most successful forms of life on Earth. Bacterial metabolism encompasses the complete set of chemical reactions that allow bacterial cells to obtain energy, build cellular components, and maintain internal stability. These metabolic processes are extraordinarily diverse, reflecting the wide range of environments bacteria inhabit. Unlike multicellular organisms with specialized tissues, each bacterial cell must independently carry out all metabolic functions necessary for survival. This autonomy has driven the evolution of highly efficient and flexible metabolic systems, capable of functioning under varying conditions of oxygen availability, temperature, pressure, and nutrient supply. One major way bacterial metabolism is classified is by oxygen requirement. Aerobic bacteria require molecular oxygen for energy production and rely on oxidative respiration, in which electrons are transferred through an electron transport chain to oxygen, forming water. Obligate aerobes cannot survive without oxygen because they depend on it as a terminal electron acceptor. In contrast, obligate anaerobes grow only in the absence of oxygen, which is toxic to them due to the lack of protective enzymes that neutralize reactive oxygen species. Facultative anaerobes can switch between aerobic respiration and anaerobic processes depending on oxygen availability, giving them a significant survival advantage in fluctuating environments. Anaerobic metabolism in bacteria includes a variety of strategies for energy generation without oxygen. Some bacteria perform anaerobic respiration using alternative terminal electron acceptors such as nitrate, sulfate, or carbon dioxide. Others rely on fermentation, a process in which organic molecules serve as both electron donors and acceptors. Fermentation yields less energy than respiration, but allows survival in oxygen-free environments. The end products of fermentation, such as organic acids, alcohols, and gases, play important roles in natural ecosystems and human applications, including food production and waste treatment. Bacterial metabolic diversity also extends to the range of substrates they can use. Some bacteria specialize in simple organic compounds like sugars and amino acids, while others can metabolize complex or unusual substances, including hydrocarbons, pesticides, and synthetic chemicals. This ability arises from diverse enzymatic pathways encoded in bacterial genomes. In many cases, bacteria are the only organisms capable of breaking down certain compounds, making them essential for decomposition and detoxification in the environment. Extreme environments have driven the evolution of bacteria with highly specialized metabolic adaptations. Thermophilic bacteria thrive at high temperatures by possessing enzymes and membrane structures that remain stable and functional under heat stress. Psychrophilic bacteria grow at very low temperatures by using enzymes with high catalytic efficiency in cold conditions. Halophilic bacteria maintain osmotic balance in highly saline environments, while acidophilic and alkaliphilic bacteria regulate internal pH to survive in extreme acidity or alkalinity. These metabolic adaptations demonstrate the biochemical flexibility of bacterial systems. Energy conservation in bacteria is closely tied to the cytoplasmic membrane, where electron transport chains and proton gradients are generated. Because bacteria lack mitochondria, all respiratory processes occur at the cell membrane. The energy stored in proton gradients is used to synthesize adenosine triphosphate, drive transport processes, and power motility. Efficient coupling of metabolism to membrane function allows bacteria to maximize energy yield from limited resources. Overall, bacterial metabolism is characterized by versatility, efficiency, and adaptability. The ability to grow aerobically, anaerobically, or under extreme conditions enables bacteria to occupy ecological niches unavailable to most other organisms. This metabolic diversity underpins the central role of bacteria in ecosystems, industry, and global biogeochemical cycles. Bacteria obtain energy and carbon through a wide range of strategies that reflect their metabolic diversity and ecological roles. One fundamental distinction is based on the source of carbon used for building cellular material. Autotrophic bacteria use carbon dioxide as their primary carbon source, synthesizing organic molecules through carbon fixation pathways. Heterotrophic bacteria require preformed organic compounds, such as sugars, amino acids, or fatty acids, which they obtain from other organisms or decaying matter. These different strategies determine how bacteria fit into food webs and nutrient cycles. Photosynthetic bacteria convert light energy into chemical energy, but their photosynthesis differs in important ways from that of plants. Cyanobacteria perform oxygenic photosynthesis, using water as an electron donor and releasing oxygen as a byproduct. This process is similar in principle to plant photosynthesis and was responsible for the accumulation of oxygen in Earth's atmosphere. Other photosynthetic bacteria carry out anaxigenic photosynthesis, which does not produce oxygen and instead uses substances such as hydrogen sulfide, sulfur, or organic compounds as electron donors. These bacteria typically inhabit anaerobic or low oxygen environments. Chemosynthetic bacteria obtain energy by oxidizing inorganic substances rather than relying on light. These bacteria play critical roles in environments where sunlight is absent or limited, such as deep-sea hydrothermal vents, underground ecosystems, and certain soil and aquatic habitats. By oxidizing compounds like ammonia, nitrite, hydrogen sulfide, ferrous iron, or hydrogen gas, chemosynthetic bacteria generate energy that is then used to fix carbon dioxide into organic matter. This process forms the base of food chains in some of the most extreme ecosystems on Earth. Heterotrophic bacteria exhibit wide variation in how they acquire energy from organic compounds. Some are obligate heterotrophs that depend entirely on organic nutrients produced by other organisms. Others are saprotrophs that decompose dead organic material, breaking down complex polymers such as cellulose, proteins, and lipids into simpler molecules. This decomposition releases nutrients back into the environment, making them available for reuse by plants and other organisms. Without heterotrophic bacteria, organic matter would accumulate and nutrient cycling would slow dramatically. Many bacteria are metabolically flexible and can switch between energy sources depending on environmental conditions. Some species can grow photoautotrophically in the presence of light and inorganic carbon, but shift to heterotrophic metabolism when organic substrates are available. Others can alternate between different inorganic energy sources as availability changes. This flexibility increases survival in environments where resource availability fluctuates and contributes to bacterial ecological success. Energy acquisition in bacteria is closely linked to electron transfer reactions, regardless of whether energy is derived from light or chemical reactions. Electrons are transferred through carriers and membrane-associated complexes to generate a proton motive force. This electrochemical gradient drives the synthesis of adenosine triphosphate and powers essential cellular processes. The efficiency and variety of these electron transfer systems underpin the vast range of bacterial metabolic lifestyles. Overall, bacterial energy strategies encompass photosynthesis, chemosynthesis, and heterotrophy, with numerous intermediate and flexible combinations. These strategies allow bacteria to function as primary producers, decomposers, and recyclers across nearly all environments. Their ability to harness energy from light, inorganic chemicals, and organic matter makes bacteria indispensable to the flow of energy through Earth's biosphere. Bacterial growth refers to an increase in cell number, rather than an increase in the size of individual cells. The primary mode of bacterial reproduction is binary fission, a process in which a single cell divides into two genetically identical daughter cells. Before division, the bacterial chromosome is replicated, and the two copies are segregated to opposite ends of the cell. A division septum then forms at the midline, guided by cytoskeletal proteins, and the cell membrane and wall constrict until separation is complete. Under optimal conditions, this process can occur rapidly, allowing bacterial populations to expand exponentially. The rate of bacterial growth is highly variable and depends on species characteristics and environmental conditions. Some bacteria can divide in less than 20 minutes, while others require hours or even days to complete a division cycle. Growth rate is influenced by nutrient availability, temperature, pH, oxygen levels, and the presence of inhibitory substances. Each species has defined minimum, optimum, and maximum conditions for growth, outside of which division slows or ceases. These limits reflect the stability and efficiency of cellular enzymes and structural components. Bacterial population growth in a closed system follows a predictable pattern known as the growth curve, which consists of distinct phases. During the lag phase, cells adapt to new environmental conditions, but do not divide rapidly. In the exponential, or log phase, cells divide at a constant and maximal rate, leading to rapid population increase. This phase is characterized by high metabolic activity and uniform cell physiology. As nutrients become depleted and waste products accumulate, growth slows, and the population enters the stationary phase, where the rate of cell division equals the rate of cell death. Eventually, unfavorable conditions lead to the death phase, in which viable cell numbers decline. Population dynamics in natural environments differ from those observed in laboratory cultures. In ecosystems, bacterial populations are rarely closed and are continuously influenced by nutrient inputs, competition, predation, and environmental fluctuations. Many bacteria exist in biofilms, structured communities attached to surfaces and embedded in a self-produced matrix. Growth within biofilms is slower and more heterogeneous than in free-living populations, but it provides protection from environmental stress and antimicrobial agents. These community-based growth patterns are common in nature and in host-associated environments. Bacterial reproduction is primarily asexual, meaning offspring are genetically identical to the parent cell except for mutations. This mode of reproduction allows for rapid population expansion and efficient colonization of suitable habitats. Genetic variation arises mainly through random mutations during DNA replication and through horizontal gene transfer, rather than through sexual reproduction. Despite the absence of meiosis and fertilization, bacterial populations can evolve quickly due to large population sizes and short generation times. Growth and reproduction are tightly regulated to ensure cellular integrity and survival. Checkpoints coordinate DNA replication, chromosome segregation, and cell division to prevent lethal errors. When conditions become unfavorable, many bacteria slow their growth rate, enter dormant states, or activate stress response pathways. These strategies allow bacterial populations to persist through periods of scarcity or stress and resume growth when conditions improve. As a result, bacterial growth and reproduction are not merely rapid but highly adaptable, supporting long-term survival across diverse environments. Bacteria possess multiple mechanisms for genetic exchange that allow them to acquire new traits independently of reproduction. These processes do not create offspring, but instead introduce genetic variation into existing cells. The four principal sources of genetic change in bacteria are mutation, transformation, transduction, and conjugation. Together, these mechanisms enable rapid adaptation to environmental pressures, including antibiotics, host defenses, and changing nutrient conditions. Mutation is the most basic source of genetic variation. It results from errors during DNA replication or from damage caused by environmental factors such as ultraviolet radiation, reactive chemicals, or oxidative stress. Mutations can involve single nucleotide changes, insertions, deletions, or larger chromosomal rearrangements. Most mutations are neutral or harmful, but some provide advantages such as improved metabolic efficiency or resistance to toxic compounds. Because bacteria reproduce rapidly and in large populations, even rare beneficial mutations can spread quickly through selection. Transformation is a process in which bacteria take up free DNA fragments from their surroundings. This DNA typically originates from dead and lysed bacterial cells. Certain bacterial species are naturally competent, meaning they possess specialized transport systems that allow DNA uptake across the cell envelope. Once inside the cell, the foreign DNA may be degraded or incorporated into the chromosome through recombination if sufficient sequence similarity exists. Transformation allows bacteria to acquire new genes without direct contact with other living cells. Transduction involves the transfer of bacterial DNA mediated by bacteriophages, which are viruses that infect bacteria. During phage replication, fragments of the host bacterial DNA can be mistakenly packaged into viral particles. When such a phage infects another bacterial cell, it injects the previous host's DNA instead of or in addition to its own genetic material. This DNA may then recombine with the recipient's genome. Transduction can transfer genes between unrelated bacteria and plays a significant role in the spread of virulence factors and antibiotic resistance genes. Conjugation is a direct, cell-to-cell mechanism of genetic exchange. It requires physical contact between a donor and a recipient cell, typically mediated by a pilus or mating bridge. The donor cell carries a conjugative plasmid that encodes the machinery necessary for DNA transfer. During conjugation, a copy of the plasmid or a portion of chromosomal DNA is transferred to the recipient. This process can occur across species boundaries and is one of the most efficient ways for bacteria to rapidly acquire complex traits. Horizontal gene transfer through transformation, transduction, and conjugation allows bacteria to evolve much faster than organisms that rely solely on vertical inheritance. Genes acquired in this way can immediately confer new functions, bypassing the slow accumulation of mutations. This genetic fluidity blurs traditional species boundaries and contributes to the extensive diversity observed among bacterial populations. It also explains how antibiotic resistance can spread rapidly in clinical and environmental settings. Dinosaur intelligence is inferred indirectly because brain tissue does not fossilize. Paleontologists reconstruct brain size and shape using natural or digital endocasts formed from sediment-filled skull cavities or CT scans. These endocasts provide information about the volume of the brain and the relative development of different regions, such as the cerebral hemispheres, olfactory bulbs, optic lobes, and cerebellum. Brain size is commonly evaluated relative to body mass, using the encephalization quotient, a comparative metric that allows broad comparisons across taxa, though it does not measure intelligence directly. Most non-avian dinosaurs had relatively small brains compared to their body size, with encephalization quotients generally similar to or slightly higher than those of modern reptiles. However, there was substantial variation among dinosaur groups. Theropod dinosaurs, especially small to medium-sized species, had proportionally larger brains than most other dinosaurs. Fossil endocasts of theropods show enlarged cerebral hemispheres and optic lobes, indicating enhanced visual processing and coordination. These features are consistent with active predatory lifestyles that required precise movement, rapid responses, and complex sensory integration. Large-bodied dinosaurs such as sauropods had extremely small brains relative to their massive size. Their encephalization quotient were among the lowest known for terrestrial vertebrates. Despite this, sauropods successfully performed essential behaviors such as locomotion, feeding, migration, growth, and reproduction over long lifespans. Their neurological organization likely relied more on distributed neural circuits and spinal processing rather than centralized brain control. Fossil evidence does not support the outdated idea that sauropods possessed multiple auxiliary brains. Instead, enlarged spinal nerve regions likely controlled limb and tail function. Ornithischian dinosaurs, including hadrosaurs, ceratopsians, and ankylosaurs, generally had intermediate brain proportions. Some hadrosaur endocasts reveal moderately developed cerebral hemispheres and well-defined cerebellar regions associated with balance and motor coordination. Large bone beds and trackway evidence indicate that certain ornithischians lived in herds, requiring coordinated movement and social tolerance. While herding behavior does not prove high intelligence, it suggests the capacity for social interaction and environmental awareness beyond purely solitary behavior. Specific brain regions provide clues about sensory priorities and behavioral complexity. Enlarged olfactory bulbs in some dinosaurs indicate a strong reliance on smell, useful for foraging, navigation, or detecting predators and conspecifics. A well-developed cerebellum suggests refined control of posture and movement, especially in bipedal species. Inner ear anatomy, closely linked to brain function, reveals information about balance, head orientation, and agility, indirectly reflecting neurological sophistication and locomotor capabilities. Comparisons with modern birds are critical for understanding dinosaur intelligence, because birds evolved directly from theropod dinosaurs. Many birds possess relatively small brains but exhibit complex behaviors such as learning, vocal communication, problem-solving, and tool use. Fossil evidence shows that some non-avian theropod had brain proportions and structures approaching those of early birds, supporting the conclusion that advanced cognitive abilities did not suddenly appear after the extinction of non-avian dinosaurs, but evolved gradually within theropod lineages. Overall, dinosaur intelligence was not uniform across the group. It varied according to body size, ecological role, and evolutionary relationships. While many dinosaurs had brains comparable to those of modern reptiles, others, particularly bird-like theropods, possessed neurological features consistent with enhanced sensory processing, coordination, and behavioral flexibility. These differences reflect adaptation to diverse environments and lifestyles, rather than a single level of cognitive ability shared by all dinosaurs. Dinosaur sensory abilities are inferred from skeletal anatomy, fossilized soft tissue impressions in rare cases, and comparisons with living relatives such as birds and crocodilians. Although sensory organs themselves do not fossilize, the bones that house them preserve reliable evidence of sensory capacity. Key sources of data include skull openings, nerve canal sizes, brain endocasts, inner ear structures, and nasal cavity complexity. Together, These features allow scientists to reconstruct how dinosaurs perceived their environment and how sensory specialization varied among groups. Vision was a dominant sense for many dinosaurs, particularly theropods. Large eye sockets and well-developed optic lobes in brain endocasts indicate strong visual processing. Forward-facing eyes in several predatory theropods suggest binocular vision, allowing depth perception important for judging distance while hunting. Some species also show evidence of large scleral rings, bony structures that supported the eye, which indicate sensitivity to low-light conditions. This suggests that certain dinosaurs may have been active during dawn, dusk, or nighttime. Herbivorous dinosaurs generally had laterally placed eyes, providing a wide field of view useful for predator detection rather than precise depth perception. Olfaction, or sense of smell, was highly developed in many dinosaurs. Enlarged olfactory bulbs in brain endocasts indicate strong scent processing ability, particularly in theropods and some ornithischians. Long and complex nasal passages increased the surface area for olfactory tissues, enhancing scent detection. A keen sense of smell would have been advantageous for locating food, identifying carrion, detecting predators, tracking prey, and recognizing members of the same species. In some predatory dinosaurs, olfactory capabilities may have rivaled or exceeded those of modern birds. Hearing ability in dinosaurs can be assessed from the structure of the inner ear and middle ear bones. The length and curvature of the cochlear duct correlate with sensitivity to different sound frequencies. Many dinosaurs had inner ear structures consistent with sensitivity to low-frequency sounds, which travel long distances and are useful for communication across open landscapes. This suggests that vocalizations or low-frequency calls may have played a role in social interaction, mating, or warning signals. The absence of external ear structures does not imply poor hearing, as modern birds and reptiles also lack external ears yet hear effectively. Balance and spatial orientation were governed by the semicircular canals of the inner ear, which are well preserved in fossil skulls. Dinosaurs with elongated semicircular canals had heightened sensitivity to head movement and body position. This trait is especially pronounced in agile, fast-moving theropods, indicating advanced balance and coordination during rapid locomotion or predatory strikes. In contrast, slower-moving, large-bodied dinosaurs tended to have shorter canals, reflecting different locomotor demands rather than inferior function. Tactile sensitivity varied across dinosaur groups and was influenced by skin coverings and facial anatomy. Fossil evidence and comparisons with birds and crocodilian suggest that some dinosaurs had sensitive facial regions supplied with dense networks of nerves. Beak-like structures in ornithischians and early birds likely contained mechanoreceptors used for precise manipulation of food. In some species, tail and body skin may also have contained sensory receptors that detected touch, vibration, or pressure, contributing to environmental awareness and social interaction. Overall, dinosaur sensory systems were diverse and well adapted to their ecological roles. Predators tended to emphasize vision, smell, and balance, while herbivores prioritized wide visual fields, hearing, and environmental awareness. These sensory adaptations demonstrate that dinosaurs were not neurologically primitive animals. Genetic exchange mechanisms are tightly regulated and often induced by specific environmental conditions, such as stress or high cell density. These systems balance the benefits of acquiring new traits against the risks of incorporating harmful DNA. Overall, mutation and horizontal gene transfer form the genetic foundation of bacterial adaptability, enabling survival and evolution across an extraordinary range of environments that live in close association with other living organisms, forming relationships that can be classified as mutualism, commensalism, or parasitism. These interactions are fundamental to biology and occur across virtually all ecosystems. In symbiosis, Bacteria occupy specific niches within or on host organisms, often engaging in long-term, evolutionarily stable relationships that influence the physiology, survival, and reproduction of both partners. Such relationships are not incidental. They are shaped by coevolution, genetic adaptation, and environmental pressures. In mutualistic relationships, both the bacteria and the host benefit. A well-documented example is the association between nitrogen-fixing bacteria and plants. Certain bacteria convert atmospheric nitrogen, which most organisms cannot use, into ammonia or related compounds that plants can assimilate to build proteins and nucleic acids. In return, the plant supplies the bacteria with carbohydrates and a protected environment. Similar mutualistic interactions exist between bacteria and animals, where bacteria aid in digestion, synthesize essential vitamins, or protect the host from harmful microbes, while receiving nutrients and stable living conditions. Commensalism describes relationships in which bacteria benefit, while the host is neither significantly helped nor harmed. Many bacteria that live on skin, in soil around plant roots, or on internal surfaces of animals fall into this category. These bacteria exploit available nutrients or physical space without disrupting host functions. Although commensal relationships appear neutral, they can shift depending on environmental conditions or changes in host health. A bacterium that is commensal under normal circumstances may become harmful if it gains access to new tissues or if host defenses are weakened. Parasitic bacteria benefit at the expense of their hosts. In these relationships, bacteria extract nutrients, damage tissues, or interfere with normal physiological processes, often leading to disease. Parasitism exists along a spectrum, from mildly harmful interactions to severe, life-threatening infections. From an evolutionary perspective, parasitism does not always favor maximal harm. Bacteria that kill their hosts too quickly may reduce their own chances of transmission. As a result, many parasitic bacteria evolve mechanisms to persist within hosts for extended periods while evading immune responses. The boundaries between mutualism, commensalism, and parasitism are not rigid. The same bacterial species can occupy different roles depending on context, environmental factors, host genetics, immune status, and microbial community structure all influence the outcome of symbiotic interactions. For example, bacteria that are harmless or beneficial in one host species may be pathogenic in another, or beneficial under certain dietary conditions and harmful under others. This flexibility reflects the dynamic nature of symbiosis rather than fixed categories. Symbiotic bacteria often undergo genetic specialization that enhances their ability to summarize relationships. In long-term mutualisms, bacteria may lose genes unnecessary for independent life and become highly dependent on their hosts. Such genome reduction is common in bacteria that live exclusively inside host cells or tissues. Conversely, bacteria involved in parasitic interactions may acquire genes that encode toxins, adhesion proteins, or immune evasion mechanisms, often through horizontal gene transfer. These genetic changes reinforce the stability and specificity of symbiotic relationships. At the ecological level, symbiotic bacteria play critical roles in maintaining biological systems. They influence nutrient availability, population dynamics, and evolutionary trajectories of host species. Without symbiotic bacteria, many organisms would be unable to survive, reproduce efficiently, or adapt to changing environments. The study of symbiotic bacteria has revealed that life on Earth is deeply interconnected, with bacterial partnerships shaping the structure and function of ecosystems, as well as the biology of individual organisms. The human microbiome refers to the collective community of bacteria that live on and inside the human body, including the skin, mouth, respiratory tract, gastrointestinal tract, and urogenital system. These These bacterial populations number in the trillions and represent thousands of distinct species. The majority reside in the digestive tract, particularly the large intestine, where conditions favor dense and diverse microbial growth. Rather than being passive occupants, these bacteria are metabolically active and play integral roles in normal human physiology. Bacteria colonize the human body shortly after birth, with early microbial composition influenced by factors such as mode of delivery, feeding practices, environment, and antibiotic exposure. Over time, microbial communities become more stable but continue to change in response to diet, age, illness, lifestyle, and environmental conditions. Each individual's microbiome is unique, shaped by genetics and life history. Yet certain bacterial groups are commonly shared across human populations. In the gastrointestinal tract, bacteria contribute directly to digestion by breaking down complex carbohydrates, fibers, and other compounds that human enzymes cannot process efficiently. Through fermentation, gut bacteria produce short-chain fatty acids that serve as energy sources for intestinal cells and influence metabolic regulation. These bacterial metabolites also affect gut motility, pH balance, and nutrient absorption, linking microbial activity to overall digestive efficiency. The human microbiome is closely involved in immune system development and regulation. Early exposure to diverse bacteria helps train the immune system to distinguish between harmful and harmless agents. Microbiome-associated bacteria stimulate immune cells, promote immune tolerance, and help prevent excessive inflammatory responses. A balanced microbial community reduces the likelihood of immune misfiring, which has been associated with allergies, autoimmune conditions, and chronic inflammatory diseases. Microbiome bacteria also provide protection against pathogenic microorganisms through competitive exclusion. By occupying physical space, consuming available nutrients, and producing antimicrobial compounds, resident bacteria limit the ability of invading pathogens to establish infections. This protective function is especially evident in the gut and on the skin, where disruptions to normal bacterial populations can increase susceptibility to disease. Different body sites host distinct bacterial communities adapted to local conditions. Skin-associated bacteria tolerate dryness, acidity, and exposure to oxygen, while gut bacteria are adapted to low-oxygen environments rich in organic nutrients. The oral microbiome includes bacteria capable of adhering to hard surfaces such as teeth, forming complex biofilms. These site-specific adaptations reflect the tight relationship between bacterial physiology and human anatomy. Disruptions to the microbiome, often referred to as dysbiosis, can result from antibiotics, infections, dietary changes, or illness. Dysbiosis has been linked to a wide range of conditions, including gastrointestinal disorders, metabolic diseases, and increased infection risk. While causation is not always clear, strong associations indicate that microbial imbalance can alter host physiology and immune responses in measurable ways. Research on the human microbiome has revealed that humans function as composite biological systems rather than independent organisms. Bacterial genes greatly outnumber human genes, expanding metabolic and biochemical capabilities beyond those encoded in human DNA alone. This integrated relationship highlights the importance of bacteria not only as external environmental agents, but as essential contributors to human health, development, and survival. Pathogenic bacteria are bacteria capable of causing disease in humans, animals, or plants by disrupting normal biological functions. Disease occurs when bacteria successfully enter the host, survive host defenses, multiply, and damage tissues either directly or indirectly. Not all bacteria are pathogenic, and pathogenicity is not an inherent property of all members of a species. Rather, It depends on specific genetic traits, environmental conditions, and host susceptibility. Many bacteria exist harmlessly or beneficially in one context and become pathogenic only when they access vulnerable tissues or when host defenses are compromised. Bacterial infection typically begins with exposure through routes such as ingestion, inhalation, skin contact, or entry through wounds. Once inside the host, pathogenic bacteria must adhere to host tissues to avoid being removed by physical processes such as mucus flow or fluid movement. Adhesion is often mediated by specialized surface structures that bind to specific host cell receptors. Successful attachment allows bacteria to colonize tissues and establish infection. After colonization, pathogenic bacteria obtain nutrients from the host environment to support growth and replication. Some bacteria exploit readily available nutrients, while others break down host tissues or interfere with cellular processes to release usable compounds. Rapid multiplication can overwhelm local defenses and increase the likelihood of tissue damage. In many infections, symptoms arise not only from bacterial activity, but also from the host's inflammatory response to bacterial presence. Pathogenic bacteria cause disease through multiple mechanisms. Some invade host cells and tissues directly, disrupting cellular integrity and function. Others remain extracellular but release substances that interfere with normal physiological processes. Tissue destruction, impaired organ function, and altered cellular signaling can result from these interactions. The severity of disease depends on bacterial load, virulence traits, infection site, and the host's immune response. The host immune system responds to pathogenic bacteria through innate and adaptive mechanisms. Physical barriers, immune cells, and antimicrobial molecules act to detect and eliminate invading bacteria. In many cases, symptoms such as fever, swelling, and pain reflect immune activation rather than direct bacterial damage. While immune responses are essential for clearing infection, excessive or misdirected responses can contribute to tissue injury and disease progression. Some pathogenic bacteria are classified as opportunistic pathogens. These bacteria normally exist as harmless members of the body's microbial communities or in the environment, but cause disease when given access to normally sterile sites or when host immunity is weakened. Medical procedures, injuries, chronic illness, or immune suppression can create conditions that allow opportunistic bacteria to proliferate and cause infection. Transmission of pathogenic bacteria occurs through direct contact, contaminated food or water, airborne particles, vectors, or environmental surfaces. Successful transmission depends on bacterial survival outside the host and the ability to reach new susceptible hosts. Some bacteria are adapted for rapid spread, while others rely on close contact or specific environmental conditions. Transmission dynamics strongly influence disease prevalence and outbreak potential. The study of pathogenic bacteria has shown that disease is the result of complex interactions between bacterial traits and host biology. Pathogenicity is not a fixed characteristic, but a dynamic outcome shaped by evolution, ecology, and host-pathogen interactions. Understanding how bacteria cause disease has been essential for developing preventive measures, treatments, and public health strategies that reduce the impact of bacterial infections on human populations. Bacterial toxins and virulence factors are molecules and structures that enhance a bacterium's ability to colonize a host, evade immune defenses, and cause disease. Virulence factors include toxins, surface proteins, enzymes, secretion systems, and regulatory mechanisms that collectively determine the severity and progression of infection. These factors are often encoded by specific genes that may be acquired through mutation or horizontal gene transfer, allowing bacteria to rapidly evolve pathogenic capabilities. Toxins are among the most direct causes of tissue damage during bacterial infections. They are broadly classified into exotoxins and endotoxins. Exotoxins are proteins actively secreted by living bacteria and typically have specific targets within host cells. They can disrupt cellular metabolism, interfere with protein synthesis, damage membranes, or alter signaling pathways. Exotoxins are often highly potent, meaning very small amounts can cause significant biological effects. Endotoxins differ fundamentally from exotoxins in structure and mode of action. They are components of the outer membrane of certain bacteria and are released primarily when bacterial cells die and break apart. Endotoxins trigger strong immune responses by activating host defense mechanisms. While they do not target specific cellular processes like exotoxins, their ability to induce inflammation, fever, and shock makes them a major contributor to severe systemic illness. Virulence factors also include bacterial structures that promote attachment to host tissues. Adhesion molecules allow bacteria to bind tightly to specific cell types, preventing removal by physical defenses such as fluid flow or mucus. This attachment is often the first critical step in infection, as it enables bacteria to establish stable colonies at particular anatomical sites. Different adhesion mechanisms contribute to tissue specificity, explaining why certain bacteria preferentially infect specific organs. Some bacteria produce enzymes that facilitate tissue invasion and nutrient acquisition. These enzymes break down host barriers such as connective tissue, cell membranes, or protective secretions. By degrading structural components of tissues, bacteria gain access to deeper layers of the host and spread beyond the initial site of infection. Enzymatic activity can also release nutrients that support bacterial growth, further enhancing infection severity. Immune evasion is a key aspect of bacterial virulence. Many bacteria possess mechanisms that allow them to avoid detection or destruction by the host immune system. These include surface modifications that prevent immune recognition, production of molecules that inhibit immune signaling, and the ability to survive inside immune cells. Some bacteria can alter their surface antigens over time, reducing the effectiveness of immune memory and prolonging infection. The expression of toxins and virulence factors is often tightly regulated rather than constant. Bacteria frequently sense environmental conditions such as temperature, nutrient availability, or host-derived signals, and adjust gene expression accordingly. This regulation allows bacteria to conserve energy and activate virulence traits only when they increase survival or transmission. Coordinated regulation enhances the efficiency of infection and minimizes unnecessary exposure to immune defenses. The combined action of toxins and virulence factors determines disease outcome. Mild infections may involve limited virulence expression, while severe disease often reflects high toxin production, widespread tissue invasion, or excessive immune activation. Understanding these mechanisms has been essential for identifying targets for medical intervention, developing treatments, and reducing the harmful effects of bacterial infections through prevention and control strategies. The immune system employs multiple layers of defense to detect, control, and eliminate bacterial infections. These defenses are broadly divided into innate and adaptive components that function together to protect the body. The innate immune system provides rapid, nonspecific responses that act as the first line of defense, while the adaptive immune system generates targeted responses tailored to specific bacterial antigens. Effective defense against bacteria depends on coordination between these systems rather than the action of any single mechanism. Physical and chemical barriers form the earliest stage of bacterial defense. Intact skin, mucous membranes, and epithelial surfaces prevent bacterial entry, while secretions such as stomach acid, bile, enzymes, and antimicrobial peptides inhibit bacterial survival. Normal microbial communities on body surfaces also contribute by limiting bacterial colonization through competition for space and nutrients. Breaches in these barriers significantly increase the risk of bacterial infection. When bacteria enter the body, innate immune cells respond rapidly. Phagocytic cells, such as neutrophils and macrophages, recognize bacterial components through pattern recognition receptors that detect conserved molecular features. These cells engulf bacteria and destroy them using enzymes, reactive molecules, and acidic environments. The innate immune response also includes the release of signaling molecules that recruit additional immune cells to the site of infection and promote inflammation. Inflammation is a central immune response to bacterial invasion. It increases blood flow and vascular permeability, allowing immune cells and proteins to reach infected tissues more efficiently. While inflammation helps contain and eliminate bacteria, it can also cause tissue damage if excessive or prolonged. Many clinical symptoms of bacterial infection, such as swelling, redness, pain, and fever, result from inflammatory processes rather than direct bacterial activity. The adaptive immune system provides specificity and long-term protection. B lymphocytes produce antibodies that bind to bacterial antigens, marking bacteria for destruction or neutralizing their ability to attach to host tissues. Antibodies also activate additional immune pathways that enhance bacterial clearance. T lymphocytes support these responses by coordinating immune activity and directly targeting infected cells when bacteria reside inside host cells. Bacteria have evolved numerous mechanisms to counter immune defenses. Some produce capsules or surface structures that inhibit recognition or phagocytosis. Others secrete molecules that interfere with immune signaling or degrade immune components. Certain bacteria can survive within immune cells by resisting destruction, effectively using the immune system as a protective niche. These defense strategies increase bacterial persistence and complicate immune clearance. The interaction between bacterial defense mechanisms and the immune system is dynamic and adaptive. As the immune system applies selective pressure, bacteria that evade detection or destruction are more likely to survive and spread. Conversely, immune memory generated by adaptive responses improves the body's ability to respond rapidly to future encounters with the same bacteria. This ongoing interaction shapes both bacterial evolution and immune system function. Understanding immune responses to bacteria has been critical for the development of vaccines, immunotherapies, and infection control strategies. It has revealed that disease severity depends not only on bacterial virulence, but also on the effectiveness and regulation of immune responses. Balanced immune activity is essential, as insufficient responses allow bacterial proliferation, while excessive responses can cause significant host damage. Antibiotics are chemical substances that inhibit the growth of bacteria or kill them outright, and have been one of the most significant medical advances in human history. They transformed the treatment of bacterial infections that were once frequently fatal into conditions that could be reliably managed or cured. Antibiotics act by targeting structures or processes essential to bacterial survival, such as cell wall synthesis, protein production, nucleic acid replication, or metabolic pathways, while generally sparing human cells. The discovery of antibiotics emerged from observations that certain microorganisms could suppress the growth of bacteria. Early antibacterial agents were derived from naturally occurring compounds produced by fungi or bacteria competing for resources in the environment. These discoveries demonstrated that microorganisms engage in chemical warfare, and that such compounds could be harnessed for medical use. The identification, purification, and mass production of antibiotics marked the beginning of modern antimicrobial therapy. Antibiotics are classified based on their mechanism of action and spectrum of activity. Some antibiotics are bactericidal, meaning they directly kill bacteria, while others are bacteriostatic, meaning they inhibit bacterial growth and rely on the immune system to clear the infection. Broad-spectrum antibiotics act against a wide range of bacterial species, whereas narrow-spectrum antibiotics target specific groups. The choice of antibiotic depends on the suspected or identified bacterial pathogen and the site of infection. The medical impact of antibiotics has been profound. They dramatically reduced mortality from bacterial diseases such as pneumonia, tuberculosis, wound infections, and bloodstream infections. Antibiotics also made complex medical procedures possible by preventing or treating infections associated with surgery, chemotherapy, organ transplantation, and intensive care. Their use extended average life expectancy and reshaped public health outcomes worldwide. Antibiotics function by exploiting fundamental differences between bacterial and human cells. For example, some antibiotics disrupt bacterial cell wall formation, a structure absent in human cells, leading to bacterial rupture and death. Others interfere with bacterial ribosomes, which differ structurally from human ribosomes, thereby blocking protein synthesis. Targeting these differences allows antibiotics to selectively affect bacteria with minimal direct harm to the host. Despite their effectiveness, antibiotics are not universally effective against all bacteria. Intrinsic resistance exists in some bacterial species due to structural or biochemical characteristics that prevent antibiotic action. Additionally, antibiotics are ineffective against viral infections, as viruses lack the cellular machinery targeted by antibacterial drugs. Misuse of antibiotics in viral illnesses does not provide benefit and contributes to broader public health problems. The widespread availability of antibiotics altered patterns of disease and medical practice. Infections that once required long-term hospitalization or resulted in high fatality rates became manageable with short courses of medication. However, this success also led to overuse and misuse, including inappropriate prescribing and incomplete treatment courses. These practices have had long-term consequences for bacterial populations and antibiotic effectiveness. The discovery and use of antibiotics revealed both the power and limitations of chemical control over bacterial infections. While antibiotics remain essential tools in medicine, their effectiveness depends on responsible use, accurate diagnosis, and continued scientific understanding of bacterial biology. The history of antibiotics illustrates how scientific discovery can reshape human health, while also creating new challenges that require ongoing attention and adaptation. Antibiotic resistance is the ability of bacteria to survive exposure to antibiotics that would normally inhibit their growth or kill them. It arises when bacterial populations undergo genetic changes that reduce or eliminate drug effectiveness. Resistance can be an inherent characteristic of certain bacteria or an acquired trait that develops over time. The spread of antibiotic resistance has become a major global health concern because it undermines the effectiveness of treatments that are central to modern medicine. Resistance develops primarily through natural selection. When bacteria are exposed to antibiotics, susceptible cells are eliminated or inhibited, while resistant variants survive and multiply. These resistant bacteria then become a larger proportion of the population. The more frequently and improperly antibiotics are used, the stronger the selective pressure favoring resistant strains. Incomplete treatment courses, incorrect dosing, and unnecessary antibiotic use accelerate this process. Bacteria acquire antibiotic resistance through several genetic mechanisms. Spontaneous mutations in bacterial DNA can alter the target site of an antibiotic, reduce drug uptake, or increase drug efflux. More significantly, bacteria can gain resistance genes from other bacteria through horizontal gene transfer. This transfer occurs via transformation, transduction, or conjugation, allowing resistance traits to spread rapidly across bacterial populations and even between different species. Resistance mechanisms vary depending on the antibiotic involved. Some bacteria produce enzymes that chemically inactivate antibiotics before they can exert their effects. Others modify the antibiotic's target, so the drug can no longer bind effectively. Certain bacteria reduce permeability of their cell membranes or actively pump antibiotics out of the cell, lowering intracellular drug concentrations to non-lethal levels. These mechanisms can act independently or in combination, increasing resistance strength. Antibiotic resistance is not confined to hospitals or clinical settings. Resistant bacteria are found in communities, agriculture, water systems, and natural environments. The use of antibiotics in livestock for disease prevention and growth promotion has contributed to the emergence of resistant strains that can spread to humans through food, direct contact, or environmental pathways. Wastewater containing antibiotics or resistant bacteria further amplifies environmental reservoirs of resistance genes. The consequences of antibiotic resistance are substantial. Infections caused by resistant bacteria are more difficult to treat, often requiring alternative drugs that may be less effective, more toxic, or more expensive. Treatment failures increase the risk of complications, prolonged illness, and death. Resistant infections also place greater strain on healthcare systems by increasing hospital stays, diagnostic complexity, and treatment costs. Antibiotic resistance threatens the effectiveness of many medical procedures that rely on infection control. Surgeries, cancer treatments, organ transplants, and care for premature infants depend on reliable antibiotics to prevent or manage infections. As resistance increases, the risks associated with these procedures rise, potentially reversing decades of medical progress. The impact extends beyond individual patients to public health and economic stability. Addressing antibiotic resistance requires coordinated global efforts. Strategies include improving antibiotic stewardship, enhancing infection prevention measures, developing new antimicrobial agents, and monitoring resistance patterns. Reducing unnecessary antibiotic use in medicine and agriculture is critical to slowing resistance development. Antibiotic resistance illustrates the adaptive capacity of bacteria and highlights the need for sustained scientific, medical, and public health responses to preserve the effectiveness of existing and future treatments. Bacteria play essential roles in natural ecosystems by driving nutrient cycles that sustain life on Earth. They are primary agents of decomposition, breaking down dead organic matter from plants, animals, and microorganisms into simpler chemical forms. Through this process, bacteria release nutrients such as carbon, nitrogen, phosphorus, and sulfur back into the environment, making them available for reuse by other organisms. Without bacterial decomposition, organic material would accumulate, and vital nutrients would become locked away, disrupting ecosystem function. In the carbon cycle, bacteria contribute to the transformation of organic carbon into inorganic forms and vice versa. Heterotrophic bacteria consume organic compounds and release carbon dioxide through respiration, returning carbon to the atmosphere or aquatic systems. Other bacteria convert carbon dioxide into organic molecules through metabolic pathways, particularly in aquatic and soil environments. These bacterial processes regulate carbon availability and influence global carbon balance. Bacteria are central to the nitrogen cycle, which is critical for the synthesis of proteins and nucleic acids in living organisms. Certain bacteria convert atmospheric nitrogen into biologically usable forms through nitrogen fixation. Other bacterial groups transform nitrogen compounds through processes such as nitrification and denitrification, regulating the balance between different nitrogen forms in soil and water. These transformations determine nutrient availability for plants and control nitrogen loss from ecosystems. The sulfur cycle also depends heavily on bacterial activity. Bacteria convert sulfur between oxidized and reduced forms, enabling its movement through ecosystems. These processes influence soil fertility, water chemistry, and atmospheric composition. Similarly, bacteria mediate transformations in the phosphorus cycle by solubilizing inorganic phosphorus and decomposing organic phosphorus compounds, making this essential nutrient accessible to plants and microorganisms. In aquatic ecosystems, bacteria form the base of microbial food webs. They recycle dissolved organic matter that is not directly usable by larger organisms, converting it into bacterial biomass. This biomass is then consumed by microscopic predators, transferring energy upward through the food chain. Bacterial activity in oceans, lakes, and rivers plays a major role in regulating nutrient availability and primary productivity. Bacteria contribute to ecosystem stability by maintaining chemical balance and supporting interactions among organisms. By regulating nutrient flows, bacteria influence plant growth, species composition, and population dynamics. Changes in bacterial communities can alter ecosystem structure, affecting everything from soil fertility to water quality. Stable bacterial populations help buffer ecosystems against environmental fluctuations by sustaining continuous nutrient recycling. Soil ecosystems are particularly dependent on bacteria. Soil bacteria decompose organic residues, form associations with plant roots, and influence soil structure. Their metabolic activities affect soil pH, nutrient retention, and moisture dynamics. Healthy soils contain diverse bacterial communities that enhance resilience to erosion, drought, and nutrient depletion, directly supporting terrestrial ecosystems and agriculture. Bacteria respond rapidly to environmental changes, making them sensitive indicators of ecosystem health. Shifts in temperature, moisture, pollution, or nutrient input can alter bacterial composition and activity, with cascading effects throughout ecosystems. The fundamental role of bacteria in nutrient cycling and ecosystem stability demonstrates that they are not merely background organisms, but key drivers of Earth's biological and chemical systems. Bacteria play a central role in agriculture by maintaining soil health, supporting plant nutrition, and influencing crop productivity. Agricultural soils contain diverse bacterial communities that regulate the availability of essential nutrients, decompose organic matter, and interact directly with plant roots. These bacterial processes are fundamental to soil fertility and determine the capacity of land to sustain long-term agricultural production. Without excessive external inputs, soil bacteria contribute to nutrient availability by decomposing plant residues, animal waste, and other organic materials. Through metabolic activity, they convert complex organic compounds into simpler forms that plants can absorb. This decomposition releases nutrients such as nitrogen, phosphorus, potassium, and trace elements into the soil solution. Without bacterial decomposition, nutrients would remain locked in organic matter and unavailable for plant uptake. Nitrogen fixation is one of the most agriculturally important bacterial processes. Certain soil bacteria convert atmospheric nitrogen into ammonia or related compounds that plants can use to synthesize proteins and nucleic acids. This process supplies a major portion of the nitrogen required for crop growth, particularly in low-input agricultural systems. Nitrogen-fixing bacteria may live freely in the soil or form close associations with plant roots, significantly reducing dependence on synthetic nitrogen fertilizers. Bacteria also regulate nitrogen transformations within the soil. Nitrifying bacteria convert ammonia into nitrate, a form readily absorbed by plants, while denitrifying bacteria convert nitrate back into gaseous nitrogen under low oxygen conditions. These processes control nitrogen availability, retention, and loss in agricultural soils. Proper balance among these bacterial activities is critical, as excessive nitrogen loss reduces soil fertility and contributes to environmental pollution. The region surrounding plant roots, known as the rhizosphere, is a hotspot of bacterial activity. Plant roots release organic compounds that attract and nourish bacteria, leading to dense microbial populations. In return, some bacteria enhance nutrient uptake, stimulate root growth, or produce substances that protect plants from soil-borne pathogens. These interactions influence plant health, growth rates, and resistance to environmental stress. Soil bacteria also affect soil structure and physical properties. By producing extracellular substances, bacteria help bind soil particles into aggregates. This aggregation improves soil aeration, water infiltration, and moisture retention, creating favorable conditions for root development. Well-structured soils are more resistant to erosion and compaction, supporting sustainable agricultural practices. Agricultural management practices strongly influence bacterial communities. Tillage, crop rotation, fertilizer use, irrigation, and pesticide application can alter bacterial diversity and activity. Practices that maintain organic matter inputs and minimize soil disturbance generally support more stable and diverse bacterial populations. In contrast, intensive practices that degrade soil structure or reduce organic inputs can diminish beneficial bacterial functions. Bacteria contribute indirectly to crop productivity by suppressing plant diseases. Some soil bacteria compete with or inhibit pathogenic microorganisms through resource competition or the production of antimicrobial compounds. By limiting the growth of harmful organisms, these bacteria reduce disease pressure and reliance on chemical control measures. The role of bacteria in agriculture highlights their importance beyond nutrient supply alone. They influence soil chemistry, structure, plant interactions, and ecosystem resilience. Sustainable agricultural systems depend on maintaining healthy bacterial communities that support continuous nutrient cycling, stable soils, and productive crops over time. Bacteria are widely used in industrial and biotechnological applications because of their metabolic diversity, rapid growth, and ability to be genetically modified. Their capacity to carry out specific biochemical reactions under controlled conditions makes them valuable tools for producing chemicals, pharmaceuticals, enzymes, and other commercially important products. Industrial microbiology relies heavily on bacterial systems to perform tasks that would be inefficient, costly, or environmentally harmful using purely chemical methods. One of the most important industrial uses of bacteria is in the production of enzymes. Bacterial enzymes are employed in detergents, food processing, textiles, paper manufacturing, and biofuel production. These enzymes catalyze reactions such as breaking down starches, proteins, fats, and cellulose. Bacteria can be cultivated in large quantities, allowing continuous enzyme production with consistent quality. Enzymes derived from bacteria often function under extreme conditions of temperature, pH, or salinity, making them suitable for industrial processes. Bacteria are central to the manufacture of pharmaceuticals. They are used to produce antibiotics, vaccines, hormones, and other therapeutic compounds. Through genetic engineering, bacteria can be modified to synthesize human proteins that would otherwise be difficult to obtain. These bacteria act as biological factories, converting simple raw materials into complex medicinal products with high specificity and efficiency. This approach has reduced production costs and increased accessibility to essential medicines. Fermentation processes driven by bacteria are fundamental to industrial biotechnology. In controlled fermentation systems, bacteria convert sugars and other substrates into organic acids, alcohols, solvents, and gases. These products are used in food preservation, chemical manufacturing, plastics, and energy production. Fermentation-based production is often more sustainable than chemical synthesis because it requires less energy and generates fewer toxic byproducts. Bacteria are also employed in the production of biofuels and renewable materials. Certain bacterial species convert biomass into fuels such as ethanol, hydrogen, or methane. Others synthesize biodegradable plastics and polymers from renewable resources. These applications aim to reduce reliance on fossil fuels and lower environmental impact. Bacterial systems can be optimized to improve yield, efficiency, and tolerance to industrial conditions. In molecular biology and genetic engineering, bacteria serve as essential research tools. They are used to clone genes, study gene function, and produce recombinant DNA. Bacterial plasmids function as vectors for introducing and replicating foreign genes. These techniques underpin modern biotechnology, enabling advances in medicine, agriculture, and environmental science. Bacteria are also used in industrial waste processing and resource recovery. They can extract valuable metals from low-grade ores, recover nutrients from waste streams, and transform industrial byproducts into usable materials. These processes reduce waste volume, lower pollution, and improve resource efficiency. Bacterial metabolism allows the transformation of complex or hazardous substances into simpler, less harmful forms. The industrial use of bacteria depends on precise control of growth conditions, nutrient supply, and genetic stability. Advances in bioreactor design, systems biology, and genetic engineering have expanded the range of bacterial applications. Bacteria have become integral components of modern industry, providing scalable, efficient, and adaptable solutions for producing materials, energy, and medicines while supporting more sustainable technological development. Bacteria play a major role in food systems through fermentation, preservation, and spoilage. Their metabolic activities alter the chemical composition of foods, affecting flavor, texture, nutritional value, and safety. Humans have exploited bacterial processes for thousands of years to transform raw ingredients into stable and palatable food products, while uncontrolled bacterial growth remains a primary cause of food deterioration and foodborne illness. Fermentation is a controlled process in which specific bacteria convert sugars and other nutrients into organic acids, gases, or other metabolic products. These transformations can preserve food, enhance flavor, and improve digestibility. In many fermented foods, bacteria lower the pH by producing acids, creating conditions that inhibit the growth of harmful microorganisms. Fermentation also modifies food structure, contributing to changes in texture and aroma that define characteristic food products. Bacterial fermentation can increase the nutritional value of food. Some bacteria synthesize vitamins or release nutrients that were previously bound in complex compounds. Fermentation can improve protein digestibility and reduce anti-nutritional factors found in raw plant materials. In certain foods, bacterial activity enhances mineral availability by breaking down compounds that limit absorption. These effects contribute to the dietary importance of fermented foods in many cultures. Food preservation relies on bacterial metabolism both directly and indirectly. In controlled fermentation, beneficial bacteria outcompete spoilage organisms by rapidly consuming nutrients and producing inhibitory substances such as organic acids. Preservation methods such as salting, drying, and refrigeration alter environmental conditions to limit bacterial growth, but selected bacteria adapted to these conditions can still be used to maintain product stability and quality. Not all bacterial activity in food is beneficial. Spoilage occurs when bacteria break down food components in ways that make food unpalatable or unsafe. Spoilage bacteria produce compounds that cause off odors, slimy textures, discoloration, and unpleasant tastes. These changes result from the degradation of proteins, fats, and carbohydrates. Spoilage does not always indicate the presence of pathogens, but it reduces food quality and shelf life. Foodborne illness is caused by pathogenic bacteria that contaminate food and survive processing or storage. These bacteria may enter food through improper handling, inadequate cooking, contaminated water, or contact with infected animals. Once ingested, they can multiply in the human body or release toxins that cause disease. Preventing bacterial contamination is a central concern in food safety systems worldwide. Temperature, moisture, oxygen availability, and nutrient content strongly influence bacterial behavior in foods. Low temperatures slow bacterial metabolism, while high temperatures can kill bacteria. Oxygen levels determine which bacteria can grow, with some thriving in oxygen-rich environments and others growing only in its absence. Understanding these factors allows food producers to control bacterial activity and reduce spoilage and contamination risks. Modern food production applies microbiological principles to manage bacterial populations. Hygiene practices, pasteurization, controlled fermentation, and packaging technologies are designed to suppress harmful bacteria while supporting beneficial ones. The role of bacteria in food highlights their dual importance as agents of both preservation and spoilage, making bacterial control essential for food quality, safety, and sustainability.